Stand clear, please. Mind the door. Stand clear.
Ayesda Bio.
博客
Ayesda Bio.
The special
Name,
Yota Kamikura
Lena Sato.
job title,
Team leaders.
organization.
HG Tokyo team.
And now I name you the winner.
We are HG Tokyo.
Today, we are talking to 3 members of the HG Tokyo team, gold medal winners of the IGEM competition, the biggest synthetic biology competition in the world.
Biology is the most sophisticated technology on the planet. Synthetic biology is how we engineer solutions for the future. And IGEMers are the heart of synthetic biology.
Jackie Manluck Yun, academic consultant. IGEM competition is like a very huge annual competition on synthetic biology, and there are many teams and high schools, and they would develop a project based on synthetic biology, but work in different fields, and they will join the competition in Paris, so once a year. And the level is actually quite high and quite pioneering. So they, even though they are just students, but they are actually making use of the most advanced technologies and knowledge.
Beginning of the project. So Lina, Ryota, Rekuto, welcome and thank you for joining us today. First of all, can you tell us what issue you were trying to solve?
Yes, we wanted to solve hay fever, or to phrase it more scientifically, allergic rhinitis. It's a huge problem in our home country, Japan, where just under half the population gets impacted. It's also estimated to create a lot of economic loss too. The current treatments are, yes, effective, but we would say that they are not enough, where they only target a small part of the cause and also are just a temporary solution. Adding on to that, there are multiple side effects. Therefore, we decided to propose a fundamentally different solution, one that does not aim for temporary symptom management or partial pathway inhibition, but instead rethinks allergic rhinitis from its biological foundations and seeks a more comprehensive long-term approach.
We are going to rewind and walk through the process. Before you agreed on what your project would be, you did a lot of research. Can you tell us what types of people you spoke to?
Other than the desktop research and the field study at our local drugstore, which definitely had a major role in the formation of our project, we also talked to specialists in traditional Chinese medicine to learn about some of the natural ways people treat hay fever, and Jabara farmers and product producers to ask about how our project would affect their industry. So we chose to work on hay fever through discussing with the members of our team about what we would like to solve. We agreed to work on hay fever because it closely resonated with ourselves, as many of us actually suffer from hay fever. We first visited our local pharmacy to see the current available solutions for hay fever, and that was when we saw the shelf filled with Kampo medicine, or traditional Japanese medicine rooted in Chinese medicine. So we talked with a specialist in traditional Chinese medicine, or TCM, and learned about chenpi, which is dried citrus peels and is used to treat hay fever. Getting inspiration from this encounter, we researched more about natural remedies for hay fever, and we found jabara, a citrus fruit mainly grown in Wakayama Prefecture in Japan. We learned about Narutin and its anti-allergic properties and started to form our idea of biosynthesizing Narutin. So then a group within our team actually went to Wakayama to conduct a field study about jabara, and they found out that people of Kitayama village, or where jabara is mainly grown, view our project of biosynthesizing Narutin as an opportunity for regional revitalization. And this is because we learned that while jabara is culturally and economically significant to the people, there's more to its therapeutic potential than what is currently known. This highlighted that our effect could play a major role in providing evidence-based validation of jabara.
You also ran an online survey. So who did you target?
One of the first surveys that we conducted was mainly about hay fever itself and how hay fever affects people's daily lives. So we first conducted a survey that targeted students at our school, as well as parents with students that attend our school. And there we were able to learn about the daily struggles that people face, as well as the struggles people face when they use nasal sprays, especially those such as how they often feel uncomfortable about using nasal sprays because of the liquid dripping down their throats. And so from there, we gained the idea of creating a new nasal spray which potentially could help alleviate these negative parts of current nasal sprays.
60% of your team suffers from hay fever. You wanted to tackle the issue of drowsiness associated to all the antihistaminic available on the market. You decided to go not for one, but two approaches. What were they?
Yeah, so our team came up with two complementary approaches to tackle hay fever. And the first approach was to biosynthesize an anti-allergic compound called nararutin in yeast. And nararutin is a naturally occurring flavonoid compound which is found in a Japanese citrus fruit called jabara, which has been shown to calm allergic responses through multiple pathways without causing drowsiness. However, jabara is a rare and relatively expensive fruit, and extracting and purifying nararutin from it is both costly and inefficient. That's why we decided to focus on developing a scalable fermentation-based production system using yeast, which could make this compound more accessible and sustainable. At the same time, we recognize that nararutin alone may not be sufficient for people with severe or persistent hay fever symptoms. So in order to address this, we developed a second approach, which was to design a cleavable multi-domain fusion peptide. And this peptide is designed to act across different phases of the allergic response by releasing functional domains in a controlled manner, which allows to suppress inflammation more precisely while minimizing the side effects.
Ayesda Bio, the specials.
And why did you select the sea jabara fruit?
So we chose citrus jabara over other citrus fruits because it has an exceptionally high nararutin content compared to commonly consumed citrus such as orange or lemons. And it's actually stated that jabara contains 13 times more nararutin content in the peels than yuzu, which is also a famous fruit found in Asia. Also, because citrus jabara is quite niche and underutilized fruit, it allowed regional revitalization for the Wakayama prefecture in Japan by spreading the value and health benefits of consuming this fruit.
What does nararutin bring that antihistamines, steroids, or immunotherapy don't?
Nararutin targets allergic disease at an upstream immunological level that conventional therapies do not. Unlike antihistamines, which only block histamine after it is released, nararutin reduces the immune drive that leads to histamine release in the first place by attenuating the IL-4 IgE axis. Unlike steroids or immunosuppressants, it does not broadly shut down immune function, but selectively modulates Th2-based allergic responses, resulting in a lower risk systemic side effects. And unlike allergen-specific immunotherapy, nararutin does not require allergen identification or long desensitization protocols, making it applicable as a general, low-burden intubation for allergic inflammation.
There is an element of environmental awareness in your project. You wanted to spread the name of the sea jabara fruit and contribute to the regional revitalization of Kitayama village. What was important to you?
When we visited Kitayama village, we noticed that many of the local shops and restaurants were centered around citrus jabara, and they were creating jams, senbei, chips, juice, and many other products to promote this fruit. So this really helped us realize how deeply jabara is interconnected to the local economy and the culture of the village. So seeing that, we felt a sense of responsibility. As a team participating in IGEM and working on a national health issue like hay fever, we wanted to help bring attention to this under-recognized citrus fruit with real therapeutic potential. By connecting jabara to biotechnology, we hope to amplify its value, not only as a local product, but as part of a solution to a nationwide problem, while respecting and supporting the community it comes from.
Let's talk about fusion peptide. Can you explain to us what it is and what you managed to achieve?
So the goal of the second project was to make a fusion peptide that works on multiple phases by combining peptides, each with effects on different phases, and utilizing the proteases released throughout these phases to manage the effects of the peptides to minimize side effects. And we were able to utilize multiple software systems to simulate things like folding, stability, cleavage, and the toxicity of the fusion peptide, and found the optimal combinations of peptides and linkers, and got the final sequence from more than 40 options.
Ayesda Bio, the special.
Precision engineering. Can you explain how that works?
Yes, so we rationally designed our cleavable fusion peptide, which is basically a single smart molecule engineered to release therapeutic components that target the first and last of the three phases of allergic rhinitis: allergic sensitization, primary reaction phase, and secondary reaction phase. And there are 4 peptides in our final design, connected all by cleavable linkers that respond to certain proteases that become abundant at nasal membrane at certain times of the phases. and therefore the linkers cleave and the peptides are each released and exhibit their effects at the right timings to reduce side effects. The third peptide, however, is a filler peptide without any effects. We initially wanted to use a peptide that inhibits eosinophil activation here, but our dry lab experiments suggested that this would lead to an unstable molecule and the linkers would not work as we want them to. So therefore, we deleted our third peptide and used a filler peptide there.
Once you achieved this, which is incredible already, you also had to think of a way to administrate the product. Once again, you addressed some social and environmental concerns. Tell us what you created.
So in addition to our biological approaches to tackle hay fever, we also focused on the environmental consequences of current solutions to hay fever or nasal sprays, and created a new 100% recyclable nasal spray. We 3D printed or designed nasal spray that avoids the metal springs used in conventional sprays, making it fully recyclable. Our model uses a pullback piston mechanism to maintain spraying function. With modular replaceable parts, the device ensures that individual parts can be replaced when a part is damaged or when more fluid is needed. So we believe that this approach extends the device's lifespan and reduces both cost and environmental impact compared to purchasing an entirely new spray unit every time.
We are HG Tokyo.
It must not have been an easy road for you, so can you tell us some of the challenges that you met along the way?
So regarding the project of biosynthesizing the compound Neurotin in yeast, one of the biggest challenges that we faced was the timeline of the wet lab project, where our initial plan was to engineer more than 10 enzymes into our host organism, which was unfeasible within the time given. Therefore, we struggled in replanning the experimental timeframe and took us more than 2 months to discuss which genes and enzymes we should select, as well as which chassis or microbial host would be best suited for the biosynthesis of Neurotin. Yet, without these 2 months of discussion and literature review, we would definitely not have succeeded in the biosynthesis of Neurotin, where our design originality lies in the strategy of combining the enzyme flavano-7-glycosyltransferase from Snapdragon with a 1,6-raminosyltransferase from Sui Orange in the yeast chassis. After all, this discussion and extensive literature review was one of the most critical steps of the whole project.
As of the fusion peptides, no one had experience of doing in silico research before. Therefore, we struggled using all those softwares that we used to decide on our sequence. Also, although the sequence itself got decided, the synthesis of the fusion peptide and the necessary regions for functional testing did not arrive on time, which delayed our planned wet experiments.
Ayesda Bio.
The special.
Now that you can reflect on this project, what would you say is your final achievement?
Our final achievement goes beyond the scientific aspects of our project and is to alleviate the sufferings of hay fever patients worldwide. Fortunately, we were the first team in the world to successfully reconstruct the Neurotin biosynthetic pathway in yeast using the key enzyme F7GT derived from Snapdragon. And we did this as a team that had not really prior background in synthetic biology. And what makes this especially meaningful to us is that we didn't just reproduce a natural compound, but we showed that a locally discovered nature-derived solution can be transformed into somewhat a global-scale biotechnology platform. And by integrating experimental results with ethical, regulatory, and social considerations, we were able to design a biotechnology-based nasal spray prototype that is not only effective, but also realistic, safe, and environmentally responsible.
And what now? What type of help do you need to take your project further and maybe see your product on the market someday?
To make it a final product and be able for humans to use them, unfortunately, or fortunately for safety, it has to undergo clinical trials. It means that it is impossible for us now to make it a product or an actual treatment. However, what we could do is to continue on with the fundamental research. We would like to perform multiple assays for each project. However, we lack both in money and facility. In order for further development, we would need more money and an institution with the required equipment that could help us.
The members of the team are
Victoria Tomi,
Ayana Mistani,
Yui Shimio,
Tako Narita,
Eri Ono,
Mei Nishida,
Marika Shimizu,
Ayaka Ohara,
Kaho Wasano,
Eita Okada, Hinata Motegi,
Hibiki Narita,
Kotaro Inui,
Maya Obe,
Maya Nakamura,
Koki Onodera,
Wakana Kichi,
Lena Sato,
Riku Tokugawa,
Ui Sasaki,
Yotaka Masato Hirozane,
Kaho Koyama,
Naoto Kimura,
Miho Tutsuka,
Yuri Takeuchi.
We are HG Tokyo.
If you want to support the team and the project, contact us at podcast@ayesdabio.com. You've been listening to Ayesda Bio.
The special.
Stay tuned for the next episode.